Thank you very much. I also wanted to add to the discussion that we've been talking a lot about using these methods for toxicity testing or mode of action or health effects. But I think another area that's related to risk assessment that these methods would be very useful for is exposure assessment and maybe dosimetry. For example, I know Christy Says has worked on a gut model and using it to assess the extent to which nanoparticles may be used in food packaging or in food additives could actually cross the gut epithelium. And hopefully Christy will say a few words about that in the discussion section. And that got me to thinking that, well, for a lot of, especially metals in soils, and then also I think some of the semi-volatile organics in soils, there's been a lot of interest in what is the bioavailability of these materials in soil. And so the research so far has developed a fairly good correlation for lead using, and bioavailability, using an in vitro solubility model as you have it. It's mainly just modeling the chemistry of the interaction between lead and the other elements in soil and minerals, and to what extent does lead in soil go into solution. But there's still a step that's missing, and that is how much lead then goes across the gastrointestinal epithelium and gets absorbed into the bloodstream. And the other thing is that particular method, although it works well for lead with like 99% correlation with in vivo in swine, it doesn't work well, it works okay, but not quite as well for other metals like arsenic. And we know manganese interacts with iron, and so absorption of manganese and iron are retarded because they both compete for the same transporter. So you could look at manganese solubility from a soil or a material like a slag, but once it's in solution, that's only part of the equation. You also have to look at how much is then absorbed into the bloodstream, given that it's competing for the same transporter. So hopefully one of these tissue on a chip type models might be useful in looking at this type of exposure. And with that, I'd like to also introduce Lauren Zeiss, who's a member of our committee and also with the California EPA. And I hope, Lauren, you'd feel free to contribute to the discussion. So with that, please, anybody step to the mic and please offer some comments and introduce yourself before you give your comments.
Thank you. My name is Jack Fowle. I'm with Science to Inform. And I think it was a great discussion. Thank you so much. Very interesting. One thing that I was wondering about, however, and if you take a look at question 2, the first part there, to what extent could you use data from these platforms in risk assessment context? A lot of the things that, based on my experience, particularly some 3 decades at EPA, is affecting that would be like the social aspects. We're here at the National Academy of Sciences, but of course, there are people who study the social sciences as well. And to be used, we heard a bit from, I believe, Dr. Weichel today, talking about it has to be accepted by the public and the transparency and so forth. And we heard a little bit from you yesterday, Wuxi, about the framework for risk assessment and the context and so forth. But it seems to me that really critical aspect is not just the technological applications, but how will people accept it? There are a number of court cases that, sort of require EPA and FDA and others to do certain things. Laws are written certain ways. Some are based on best available technology, but some are based on benefit-cost trade-offs, et cetera. Also, if you don't have the Office of Management Budget, you don't have the Congress, don't have the President accepting them, or public interest groups, NGOs of various sorts and so forth, feeling that these approaches are as safe, if not more informative and safe, they simply won't be used. It'd be like bringing this beautiful feast to a table. And you would expect to see, silverware and crystal and so forth, but maybe there's a picnic bench there or something. It's not going to fit. It's not going to work. So how do you make sure that the social aspects are dealt with as these tools and techniques or so forth are being developed, so when you bring the feast to the table, it's set properly? Anybody care to comment on that? I guess what I asked for. Because it seems like that's a big part. To me, I think that's going to be the biggest challenge. And I wonder if people could react to that or offer some comments, please.
Do you want to?
Excuse me.
Yeah, I think that the way I would...
Please introduce yourself for the folks
Lance
on the
Taylor,
webinar.
University of Pittsburgh.
Thank
The way I would respond to that is, I think it's accepted fact that the present gold standard in testing is animal testing. And I referred to it as not the gold standard, but the bronze standard. I mean, we know in some organs it's 50 to 70% predictive of human toxic liabilities. So I think for the public to accept it, as well as regulatory agencies, we have to demonstrate that these new tissues on a chip or organs on a chip are at least as good as animals, and the expectation is that if we optimize the tissue engineering using the right cells in the right organization with the right complexity, the theory is we can do better than animal testing. That's going to take some time, and there has to be a correlation with the existing, quote, gold standard.
Yeah, if I might interject, I think given that we're already using in vitro methods with cells and just glassware in the lab, but again, I think the difference now is we're going to hopefully move to the point where we can have just the tissue chip methods being used rather than doing the full animal study. I think it will be appealing to the public in that I think there is great public pressure to reduce the number of animal studies and use of animals, as in Europe, maybe not quite as much pressure, but I think that part will be appealing. And then I guess it'll be a matter of being able to explain the technology and showing that it does work, and you have good confidence in it.
Thank you. This is Jack Folligan. I'm sorry, maybe I miscommunicated a little bit. I agree, it's really important to show the data and show how important it is. But oftentimes, people react with their hearts, not their heads. And yes, while there are some NGAs and so forth who are really interested in reducing the use of animals, there are others, particularly those concerned about endocrine disrupting effects, don't care if you killed every last laboratory rat and animal in the whole world. They want to protect babies. They don't really care about that so much. So how do you sort of raise, not only just bring in the facts and show that it's more as informative, it's more useful and so forth, but what steps are being taken at EPA and FDA and others, or are there? What are the discussions at this point in time now to try to get to the hearts as well as the heads and roll it out so you're dealing with the social aspects as well as the technological aspects? That's what I was trying to get at.
Yes, go ahead, please.
I think a key aspect is manage expectations. We certainly, certainly there's a lot of promise in technology and these platforms and the devices, but we don't want to oversell it. We don't want to overpromise what we can deliver at this point. And I think that is a key in terms of social acceptance, certainly both in terms of scientific as well as the general community. Our leaders at the NIH would typically go to Congress and get appropriations for supporting programs like this at the NIH, and certainly in this past year has been, the tissues and chips have been cited as examples of successful programs, but yet we try to manage and say, this as exciting as these technologies can be, there are still limitations. There are still things that we need to demonstrate before we can even say they can replace or supplement animal testing. Lance is correct that there are certain circles within the community that says we have to do away with animal testing. Yes, there are ethical and social issues that would push for that. This is, we're not saying that this is the device that will do that. It could help, and certainly have been in conversations with a number of organizations, the NC3Rs and Humane Society. But again, that is not the goal. Our goal is to put forth the best science as we could and demonstrate that the technologies we're pushing forth have their own inherent limitations, and we have to acknowledge what those limitations are.
I think I think another couple of ideas on your question. One is that, so now in the IRIS program now we have we have these early engagement meetings with the public where when we're starting an assessment, before we even start assessment, we have sort of we're developing these problem formulation documents, and this is part of what came out of the NRC report from a couple of months ago, to sort of do problem formulation, understand what's out there, and if there's this type of data, we bring that to the table at the very beginning, so that people are aware of it. We can get public input and get people aware of what kind of information is out there, what kind of technologies, et cetera, and have given them an early opportunity to engage and continue to engage throughout the process. So I think that one part of engaging with the public is sort of early and often engagement. A second is maybe on a more community level, if there's applications essentially where there's nothing else, and if this can show some value added in where there's a clear gap, maybe something like the West Virginia thing earlier this year, or some sort of situation where there's no other information out there, and you can show not to overpromise, but to that this at least provides some additional information that you wouldn't be able to get any other way. That can sort of build some confidence, and you would want to pick those situations carefully to make sure that you're not setting yourself up for failure, because that could have the opposite effect. But kind of showing, being engaged at the community level, I think would also be another potential path.
So if I could add to that, I think one of the things that would be a mistake is to let these different types of technologies be evaluated independently, as opposed to being evaluated collectively as a collection of alternative methods that can be used to potentially replace animal studies. Because you can pick at the pieces, and the whole ship will sink. But if you put together a cohesive battery of information, and tissues on a chip is one aspect of a larger set of studies, a larger set of approaches that collectively can have a better predictive value against animal studies or against what we know in humans, rather than having them, as I said, pick it at the individual ones, and then they tend to fall apart more quickly.
Thank you.
So if
Helmer? I'm sorry, did you want to add something? Sorry,
I
go ahead,
would
Frank.
like to add it. There are many excellent thoughts, but I wanted to also build on some of that actually was said, and that's in the context of perhaps the at least for the part of the FDA, in terms of the responsibility of regulatory agencies in the context in this dialogue. And we all, I believe, agree it is a dialogue. It is a dialogue about striking and defining a balance. And yes, you can have proponents and opponents of animal experimentation and using animals as indicators for conditions that are compatible with life and as such. However, at the end, we have to define our, if you want to call it biosphere, globe right now. We live on this earth, and we have to understand that we are all involved in defining pathways and approaches for the future to be able to actually live here. And as such, we will have to have a dialogue about how to do this. In principle, and I think it has been articulated many times, the agency believes that regulatory decisions have to be science-based. They have to be based on information and facts, and certainly they are always influenced by our passion and by our moral values. As such, even here, it's not just that the facts will speak for itself. However, I think it does help to understand when we talk about systems that detect, let's say, environmental challenges, that we understand the sensitivity of an assay system, that we understand the precision with which it measures, and also understand the accuracy in which it translates into a meaning to a human creature or any other living creature on this earth. So I think those kind of things are important to combine, form cohesion, and perhaps if you want convergence between the fields. It has been, in the past talked about convergence, and it's kind of a new trendy word to use, but I think there's something in it that we need to understand in terms of forming a social dialogue about it, and I think it's an acceptance that's based as much as on facts and scientific evidence, as much as it is based on our fundamental beliefs, our values, and our core values as human beings.
Thank you. Go ahead, Helmut.
Okay, so Helmut Zarbel, Rutgers, a member of the committee. So I share the feeling that it's really important to get buy-in from the stakeholders and the community and so on. But I'd like to take a step back and ask the question, what is it going to take to get buy-in from the regulators? How do you satisfy ICVAM? How do you satisfy REACH? How do you satisfy the FDA to make this acceptable? What are some of the steps that are going to need to be taken before this can actually have a regulatory impact?
Well, as one of the program leaders on this project, we had taken the steps to engage the FDA at the very get-go. So DARPA and NIH are funding partners, but we do have a partnership with the FDA as well in this program to make sure that our investigators are interacting with FDA experts and making sure that the process of developing and improving and the kind of data that we need to generate are essentially things that the FDA can look into and certainly for use in regulatory decision making, that those are tools that would qualify these devices. I mean, there's the aspect, as I mentioned earlier, of efficacy, which doesn't necessarily require FDA approval. I think that by itself could be a scientific achievement and will essentially, the chips will stand on its own based on use as efficacy models. But as in terms of regulatory decision-making tools, I think the engagement with the FDA is a critical path that we had acknowledged early on, and it's now really been a crucial part of the program. And it will be much more so in the next 3 years of this program.
I was thinking more about validation.
Well, yeah, so in terms of validation, we have, our interactions, certainly I'll let Frank speak for himself, but in our interactions with the FDA, it's hitting these devices with as many compounds that we know with known effects and showing essentially how it is equal, if not superior, to existing technologies or existing assays. And so it's really more of looking at the collective body of data that's coming out of these tissue chips and presenting that to the FDA and saying these are the context of use that we're proposing to use this tissue chips, and this is the body of data to support it. And I've heard as many as 10, 20 compounds at least, and certainly our investigators are already looking at even 50 to 100 compounds. So I think we're on the road to getting it hopefully qualified in the near future, but we'll wait and see.
Let me just follow up on that, if I can, Ivan Rusin, member of the committee. So Frank, I was wondering, what can you tell us about the experiences that the agency had with seeing data packages coming in with some of these newer technologies that are really not your traditional 2D hepatocytes or some other simpler models? Has the agency had any experiences, and what has been the reaction, if you can share that with us?
Certainly there is new technology coming to the agency, and also in the context of tissue and cells on a chips in a chip system, and in that context I think there's a lot of exciting work going on. Now, in principle, you may be aware of the fact that in the context of validation of drug development tool and biomarkers, I think the agency has outlined a general strategy as to how to deal with it. And a lot, Dan, you mentioned that already, has to do with defining the purpose and the specific context of use, because it is unrealistic to expect that the agency can write out a blank check for any kind of new technology just because, let's say, it involves some nanomaterial or something like that. And that certainly is logical and rational. So I think it's in many cases the definition in dialogue of a specific pathway that will be chosen. And if you talk about test and test systems, I think one has to define what the purpose of this particular system should be, at least at the beginning. Is it a point of care, a diagnostic test? Is it a companion diagnostic in the context of another treatment development? Or is it a esoteric laboratory-based test that will be only performed in one or the other specific laboratory? And I think with that, it indicates the openness and the willingness of the agency to consider different approaches into different directions. Again, it's a question of dialogue. I think we have a responsibility towards the public to ensure that we do not misrepresent the situation, that the public has confidence in the safety and the validity of the test systems that are used. And as such, we will have to define the specific pathway for one or the other approach. But I think some of the critical elements, they were just mentioned here, and I think it should provide some guidance.
So
Bill.
if I could follow up on that, sorry, Bill. You have to stand there a few more minutes. So with the genomics era, there was the concept of that FDA would have a safe harbor, and that information that might be submitted along with drug applications that was new would not necessarily be held against the compound because it was an experimental approach. Is it the sense that there's a lot of tissue-on-a-chip information being generated that never sees the light of day because there's some reluctance to share it? I don't know. I'm asking.
Well, I think it's in one way it's a very philosophical question, but on the other hand, it's a very practical question too. Philosophically speaking, I think it is a matter of fact that we seem to be in an era where we have a crisis of trust. So we need to deal with this as partners and have adult conversations about it and talk about the facts. But philosophically, I would state, and again, that's my personal perspective, that information that is generated and provided in a context of a specific approach, a specific test, a system on a chip, is not used to the disadvantage of either the sponsor or the, if you want to think in a bigger frame, the public. Because knowledge does help. And again, that's a philosophical question that I would make here, a philosophical statement. And as such, that what we know can help to guide into a better direction and realize opportunities, recognize them. And that what we know also can help to prevent failures and perhaps, let me call it mistakes or limitations of systems. And I think we should and we will use this knowledge responsibly.
Just also to add context in terms of what the steps are being taken. So we, as I have said, the status of the field right now is these are primarily standalone organs on chips or tissues on chips. And so certainly while the NIH has made available to our investigators testing and training compounds as well as validation set of compounds, 100 or so or more, and we're still in the process of negotiating with large pharma, additional pharma to add to this collection. This will be an iterative process. So as two or more organs are being linked together, you know, more and more of the compounds will have to be tested and have to be retested so that we know that during the linking process of the tissues on chips or organs on chip, none of the phenotype or the characteristics that we desire are being lost. And so this is not something that we're going to have a a fixed data set that will be opened up. So I think this is there will be a lot of iterative involvement in terms of how to determine how predictive these models are going to be.
Okay. Bill.
Bill Farland, Colorado State, member of the committee. I guess I'd like to bring us back to the discussion about human variability. And three of you mentioned human variability and opportunities for these platforms to help us with that. John talked a little bit about the confidence that we might have in chemicals. And of course, that brings in the question of sensitive individuals as well as you screen chemicals. There are really two aspects to this. One of them is that the tissue chip, as we've talked about it, is really an N of 1. And Cheryl yesterday raised the question of gene-environment interactions and the fact that the epigenome may not be as well represented in the chip as the genome is once you go through the IPCS process of differentiation. So I guess the first question has to do with how do we approach this in terms of informing variability in the human population and understanding how these represent an average or a sensitive individual. And then the second part of this, I'd like to just reinforce something that Wei Shui said, and that's that we've made great progress in starting to think about pre-existing disease. And the ability to use these chips as a way to test chemicals against a system that essentially is compromised is representative of somebody with liver disease. We know from chlorinated compounds that people with alcoholic livers have greater sensitivity for tissue damage. We know that for metals, individuals with perturbed renal function essentially have greater impacts of metal exposures because they don't clear them as well. How do we deal with some of those issues and take advantage of these technologies to address that?
Anybody care to address the individual susceptibility topic?
Well, I think the second thing you said is in part answer to the first in terms of genetic variability is probably only a small fraction of the overall variability across the population. And it's really maybe this gets back to the exposome workshop, right? So how do we reproduce the exposome in a tissue chip to have sort of a realistic, aggregation of perturbations and exposures over the life of the chip, I guess. And, being able to make progress there could really, open up a whole new range of investigation for human variability and susceptibility. But we have to walk before we run, or maybe crawl before we walk. So.
Yeah, so just in terms of representing genetic variability, I know there are already efforts within the community in terms of collecting upwards of 15,000 fibroblast lines from individuals who are in the process of doing whole genome sequencing on this. So in some ways you can have certainly a good sense of the pharmacogenomic profiles as well as even the epigenetic information going in. And certainly once there's also a partnership to transform those fibroblast line into IPS lines. And so at some point, once all these IPS lines are available and can be seeded into these platforms, I think that's where we can see and realize the studies in terms of variability. one can also look at taking known genetic polymorphisms, and let's say for a complex disease, I think there was a lot of talk that the potential of these devices can be, so like the lowest hanging fruit would be on single Mendelian disorders. But I would argue that you can also look at a complex trait, put them on an isogenic background, and slowly tease out what are the contributing factors for that complex trait. So there is a lot of promise. There's a lot of potential. I think it's just being right now the state is just to look at certainly a few select IPS lines, and because we're still in the proof of concept stages and trying to validate this platforms.
I think another challenge in terms of trying to do this is being able to sort of accelerate the time course, because we don't want to have to wait 70 years for a tissue chip to develop chronic conditions, right? So we need to somehow both recapitulate, there was talk yesterday about whether the flow rates are equivalent, so it's sort of this conundrum. We want to recapitulate the human, on a micro level, but then we want to accelerate the aging process in essence, or the lifetime accumulation of insults to be able to reproduce the general population susceptibility. So I think it's a difficult question.
Yes, and to add to this, of course it's a difficult question, otherwise we wouldn't be puzzled by it, but I think it's an iterative process, and I think what has been done and what we are trying to do with on the micro level and the organs on a chip direction should and has to be met, and if you want, it has to be compatible and compared to, again, I say it, real-time physiologic processes that we need to be able to understand better. And I think for society, we have to make significant investments into this. And I'm saying that also because it's not just specific clinical outcomes the way we measure it right now. We have our clinical endpoints, and sometimes we struggle to define them, particularly when we want to define the effectiveness and the utility of a new drug. To define exactly what is the clinical change that we are measuring here that indicates that there is something that is desirable. But it also has something to do with our ability to measure our daily state of well-being. And I think here is where technology helps us in some ways, but I think at the end it's still a judgment that we make as human beings and as a society. How do we use information that's becoming available through our personal devices that we are carrying around that can monitor our daily state of well-being and give that information into a meaningful set of data and combine it with other information that our, let's say, blood test will reveal, or a whole body scan in many ways. And I'm talking a little bit futuristic, but I think this is the direction it has to go. We have to validate microsystems and in vitro systems through physiologic processes, and we need to do both. We cannot just invest into one direction.
Thank you. John, were you first or May?
John is first.
Okay, good. John.
I think I may have been. John Balbus with NIEHS. So I'm going to ask a question that's a little bit of a follow-up to Bill's, and also maybe even to Jackson. And I'm really struck at the analogous situation that we were in 10 years ago with toxicogenomics in terms of all of this potential and this belief that we're going to be able to represent a lot of biological diversity, and then the challenges when we actually try to do that. So I want to focus in on one thing, which is what I think is a trade-off between an attempt to represent the true diversity in human genotypic and phenotypic states for whatever given problem we're looking at versus reproducibility. And I'm just wondering, from your perspectives, do you see this, and also it strikes me, and again, I'm not the expert in this, but it strikes me that the process of creating these chips, certainly in the present time, has a certain degree of stochasticity, because we are relying on a differentiation process that has its own self-regulating and actuating state, and there are going to be variations in that, micro variations in that, the epigenetic variations in that as that goes on. So there's, I think, some inherent variability in the creation of these chips that both contributes to the depiction of human diversity, but also, I think, impairs the reproducibility of the system. So do you see this as, that we're going to be going to a state where we have thousands of chips for every given assay to try to represent that human diversity? Or do you think it's a question of controlling very tightly and getting much more insight into the process so that there is much tighter control? Do you see it as being solved by a statistical process or by a real hands-on understanding of exactly the nature of what you have on every individual chip and being able to reproduce that and create that again and again? And is that a question that makes sense?
You've stumped them.
Well, maybe you need some of both, because I mean, twins start off the same, but then there's obviously some stochasticity that leads to that they do, diverge.
But this is an assay that differentiates itself, and so it has
exactly.
that aspect to it.
Right. So essentially, if you have 1,000 of the same genome, then you have 1,000 twins. Each of which there'll be slight variations. But then you also want, it's hard. I think, depending on the purpose of the investigation, you might want one or the other. So, maybe that's the fit for purpose kind of idea. But you might, from a scientific, hypothesis testing, et cetera, you want to reduce variation as much as possible, except in the thing that you want to vary, right? But then to apply it to the real world situation where everything does vary, then maybe you want to introduce some of that back in, so.
That's the trade-off, right?
Right.
Yeah, it's really no different than what we're doing in tox studies right now. We use, to identify a potential unknown toxicant, we use inbred animals or as closely as controlled as possible. And then to look at the range of human variation or the range of animal variation, we use something like the diversity outbred animal, and then where you can actually incorporate a range of diversity that's wider than the human population, or thought to be wider than the human population, and actually use those animals to be able to identify the genes that are responsible for that are driving that difference in response. So it depends on what you're asking.
Right. I think the other aspect that, of the diversity outbred and those is that is reproducible variability. So it's both variable, but it's also reproducible. And so I think that is sort of the key to threading the needle. So you want to have the variability, but you want the variability to be reproducible, each time you run the experiment. And not for, if you use, just random outbred or, just random samples from a population, different random samples each time, then you're going to get a different variability each time. Whereas if you have a sort of a controlled battery of variability, then that might be, both scientifically reproducible as well as addressing the variability.
Thank you. I think May was next.
Okay, so my thinking is that this is, I thought back, many years ago, I chaired this high throughput screening, whatever, initiatives. It's the same thing. We are very excited about this possibility of robotic, screening of thousands of chemicals and, understanding the mechanisms. So as long as we actually can acknowledge the limitations of this particular system or this set of systems, and not to claim that it's going to really be able to, because this is a model. So once we identify the limitations, then we can actually put the data that we collected in perspectives. So no models can totally reproduce what human situation is. So we do not need to go extensively, at least at this stage, to try to defend the system, because it is a novel technology. However, it has lots of limitations. So we should perhaps spend some time defining those limitations. So that's point number one. The second point I really want to make is that I feel that it is important as we selected these cell lines, we really address the issue of health disparity. So I don't know how you select the cell lines, but it's important that we cover a range. And the final question I have is, because these technologies are so expensive and so difficult to do, I think it would be important for the agencies or the, either NIH or other regulatory agencies to support research that is outside all these big farmers' platform. Because I do remember in the past when we do robotic screening, not many places can reproduce it. So it's important to really promote grassroots type of research. And those are the 3 points I'd like to make. And this is Shuk Mei-Ho, University of Cincinnati.
Thank you.
Member of the committee.
Anybody want to make a comment? Good points. I think Anna?
Yes.
Is that right?
Ann Bostrom, University of Washington, member of the committee. And actually, my first question follows on to May's point. You mentioned, Danilo, that you have involved FDA early on in this grants program, and that's commendable. It's really great to see it, and it's obviously having some effect. Is there a possibility, given, despite the organizational challenges, of involving EPA equally with the investigators?
No, so we're certainly open to partnerships, and as the in the inception of the program, we identified the key stakeholders, and certainly identified FDA as one of the key ones. Certainly in this second phase of the program, we are expanding that partnership, and that's why I had mentioned that we are in conversations right now with leading pharmaceutical companies to expand our collection of compounds. And they are making available not only the compounds, but the associated preclinical and clinical data that will further lead to further validation of these platforms. So we are prepared to expand. Again, it's part of our managed expectations in terms of wanting to demonstrate first what we can achieve with these platforms, and then essentially the utility can be expanded, whether it be for looking at exposomes and through the EPA and other agencies. I think those are the kind of things that we just gradually build up into the program. This is not a program that will go away in 5 years. I suspect that this will have a life on its own, and if I have my way, I intend to pursue another 5 years of the program with as I've said, the future, including clinical trials on chips, and hopefully be able to bring in additional partners in the next iteration of this program.
Excellent. And I think, as Wei-Shui said, early involvement of stakeholders is really critical and can shape what's happening. And so I'd encourage you to get involved with EPA more closely sooner rather than later. My second point follows on a point that Wei-Shui made, which is a question about computational toxicology. And this really goes to the investigators who spoke yesterday more than to you all, but it also seemed from a risk assessment perspective that this idea of meshing computational toxicology and analytical approaches to modeling with tissues on a chip is really promising and is important, and I'm wondering if you're doing it. Is anybody doing it?
Yes.
Oh, yeah. Great.
Hey, Lance.
So, yes, we think it's critical.
Please give your name.
Lance Taylor,
Yeah, I know.
University of
Thank you.
Pittsburgh. So inherent in what we've been doing, and now we're going to be making it available to all the investigators, is the development initially of a microphysiology database for a tool that can collect this complex data of multiple parameters over time, and with lots of variables as we develop the systems, changing the cell types, looking at stem cells differentiated in different ways, different lots, collecting all of that data, and then having it be available for computational analysis. Because the ultimate goal is to make a predictive tool where you can have the kind of data from an analytical system that warrants making predictive tests. So one of our goals is to have enough compounds through the device once it is stable and more than a advanced prototype, to generate a data set of 1,000 compounds of known toxic liabilities, and then see how well the system does relative to animal data, which we can also access and do the predictions back and forth. I think that's critical. Just having the analytical data, publishing papers, and having spreadsheets of data is not worth doing from our perspective. So we think that's a critical. While I'm here, I want to make one pleasantly encouraging question, and that gets to the transparency that was raised, which I think is good. It's clear that we have to, quote, demonstrate the science and then, quote, sell the results relative to the present standards. The question I would ask, in a pleasantly provocative way, does the public really know how limited animal testing is, which is a key reference for the FDA? In other words, we know that gold standard isn't really gold, and so we're trying to kind of develop a platform that is better than the limitations of animal testing.
Well, since you mentioned FDA, I guess I would like to try and address that. I think it depends, of course, to what extent the individual is interested and involved. But in general, I think the research community does recognize broadly that predictability from animal systems and the limitation therein, and also we are all quite aware, any one of you and any one of us who deals with clinical development, it's a big challenge to take the step from the non-clinical into the clinical, and there is a world of unknowns when you make that step, and certainly anyone who thinks about it should be aware of it. But you brought up a very important point, which I think is a great opportunity, namely to show that microphysiological systems or systems on a chip and the in combination with the information accumulated and analyzed provides an advantage over the relative imprecision of animal systems to predict human beings. And I think this is something where we will find more and more evidence for, and I think this is something important that we need to continue to communicate to the public.
Thank you. Rosemary?
Hi, Rosemary Hunsaker from NIH. I think I'm asking a question that Anne has just asked, but I'm going to ask you, I'm going to put you on the spot and ask you for a more definitive answer. So I think the reason that the DARPA NIH program is getting so much traction is because there's a very real customer base out there that's critically asking for these tools, and that's the pharma developers who really want to use this for making early decision making before they even get to the point where they're ready to start considering approaching the FDA. You know, there's a parallel track that's developing some of these tools that may be able to be submitted for new biomarker consideration, but that actually is a separate track. It's the same sort of general path, but it veers off a bit. So the question I have is that as we develop these tools, because we're sort of focused on pharma's needs right now in the NIH and DARPA program, do you panel members see, after what you've heard over the last day or so, any reason to want to put the brakes on? And I don't mean in the development of the systems, but are there ways where these systems that we know can be commonly used in some ways for both drug development and environmental toxicity assessment? Are there things about them that you've heard that give you red flags that say, wait a minute, we need to take a little bit of a step back to be more broad in our development so that we can make sure that we don't shut down anything in these development pathways that would make the tools accessible and usable for a broader environmental assessment?
Well, okay, let me try and get it started.
Fred. Frank, yeah.
Yes.
Sorry.
I don't think that anyone at FDA is considering shutting down one or the other pathway. I think it's really a question of resources and prioritization, and I think in a sense also where do we think we can make the greatest impact, because we are at the critical phase where the investments that we have made, we want to justify them. And I think the pragmatism that is driving some of the development in the different systems is understandable. I do not believe that it is specifically pharma-focused. I think that certainly pharmaceutical industry is a significant part of our socioeconomic environment, if you want to think about it that way, and certainly is an important force that can leverage investments and also justify some of them. But at the end, it's boiling down to being pragmatic, defining where can we make the biggest mark at a time to make some progress which will form the next foundation to take the next step. At least that's how I see it.
Thank you. So let's take one more question and response cycle. Oh, go ahead.
Yeah, thank you. So just to add to what Frank said, and perhaps address May's third point in terms of accessibility, I think certainly the key, and not so much as putting the brakes on it, is just to make the devices, the technology, as accessible as possible, and as certainly as quickly as possible. So we're not, we built the program not necessarily just to address pharma needs, but to develop this as a scientific tool. And so in light of that, we have actually taken the steps, the NIH has taken steps to issue out a request for information from biotechs and other entities that might be interested in commercialization of these devices, albeit right now on standalone devices, standalone organs and chips. And so our goal is to get into the hands of basic researchers, preclinical investigators, clinical investigators, who will then hopefully put it into different applications, whether it be for looking at exposure biology or other potential use of these devices. So I think that there are plenty of creative and innovative scientists out there, so you just get the tools into their hands, and I'm sure they will think of innovative ways of putting them into good use.
Thank you. So let's take two more questions, commenters. I sense we're starting to morph into the next topic, which is infusing regulatory considerations into research and development, and we have a break coming up. So let's take two more questions, comments, and then we'll take a break, and then we'll really get into it after the break.
Great.
Thank you.
Thank you. That sounds good. I'm Barbara Cleaforth with the EPA's Office of Research and Development. I have really more of a follow-on comment to Ann's comments. I'm with the Chemical Safety for Sustainability program at EPA, and we do have some, we are definitely hoping to leverage our resources and collaborate more with NIH and others, all our varied stakeholders out there. It is a little difficult to find information and hard data on a lot of things, as you all know, but we do have a series of RFAs that we're getting ready to award this fall, as well as a number of in-house projects. The ones we're awarding this fall are on organotypic culture models for predictive toxicology. We have some investigators in the room, and we do hope that to be an ongoing series of RFAs or RFPs to, and hopefully we'll work closer with NIH and our program colleagues at EPA to further refine those, because this field is moving fast, but we, leveraging and getting more data into EPA is what we're all about. That's all I have to say.
Great. Thank you. Lauren?
Hi, Lauren Zeiss, a member of the committee. Just a few points. One is, I think we've heard a number of times this issue of gold standard, and then heard about looking at a series of animal studies or animal outcomes and comparing it to what comes out. And I really do think we need to step back and think about what we really are talking about, given we have these human cell based devices, what should be the gold standard? I think it's a question. And I don't know if there's been further discussion maybe at NTP or FDA about that issue, so that's a question. Then on the whole variability issue, we've been talking a lot about within the risk assessment community of developing these distributions of variability across the population. It's useful for doing things like benefit analyses, because you can get a better estimate of what the true benefits are. But it seems in some regulatory contexts, what we really need to know is, well, what's the most sensitive group that we really need to pay attention to? And it seems that these systems have a lot of promise. Take the metals example in the kidney, for figuring out maybe a series of inputs into the device to kind of come up with sort of the most sensitive. And so from a quantitative point of view, getting a better handle on what's a reasonable uncertainty factor. And then the final thing is, I think we're talking about very different regulatory paradigms in the sense that using these devices in a pharma context for sorting through chemicals or even bringing packages to the FDA is a different world from the EPA world or the Cal EPA world, where for many chemicals there's not a lot of data, and you're kind of stuck looking at what you have available to you. And one of the questions is when we could even take some experimental outcomes coming out of the development process, looking at some outcomes that may signal a potential problem, and maybe use that as some information that helps us make a decision about an environmental chemical. And yesterday we heard an example of a widely used chemical that was a follicular toxicant. And so, that information I don't think was around at the time that EPA was thinking about dispersants, and it was a dispersant for oil dispersant. How much attention should be placed on those kinds of outcomes, and when are they ready for that kind of use? I think it's a different setting than a regulatory approval for a pharmaceutical or something like that.
Mm-hmm. Thank you. Great points. Anybody care to comment?
Well, just on part of your question, Lauren, and the gold standard issue is one that comes up all the time. And as Associate Director of the National Toxicology Program, is sort of like Nixon going to China, right? Obviously, we have an issue that we have a large body of information that I almost said undermined, is the foundation for a large amount of regulatory activity. But in the NTP, we also have programs that for our report on carcinogens and our Office of Health Assessment and Translation that make a very strong effort to try to pull together evidence from all different types of information, human epidemiology, animal data. And we're spending an awful lot of time trying to understand what I was getting at earlier, the confidence that we can have in mechanistic information, and the kind of information that comes from tissues on a chip is just sort of one step removed from mechanism or mode of action information, at least in the way I see it. So the animal studies they have to be remembered as being part of a larger body of information. So when we put together databases of chemicals that you want to run through tissues on a chip, make sure that you're picking chemicals that have a cohesive body of information so that you're not stuck with an animal alpha-2U kidney carcinogen that is probably not relevant for humans. So, I mean, just be cognizant of that when you're putting these databases together. And I think that's going to get around this issue of well, the animal data aren't any good, they're only 50% predictive, et cetera, et cetera, et cetera. That's not really the right question or the right point, I think, to make.
Yeah, I liked Wei Shui's, I think, initial comment that this is part of a body of evidence that will hopefully improve our ability to better predict variability and be protective of the sensitive endpoints for the population. Anybody else have any comments related to that before we take a break? Go ahead, please.
Well, if I may just add to this, I think the question of gold standards towards and having human well-being and health in mind in the first place certainly is a very reasonable goal to have. But you know, if something is bad in an animal system, it's certainly not good for our environment, I can tell you that. So I think it's the complexity of data that has to be seen in context. And certainly before we make decisions to disperse something that's harmful in rodents into a broader sense in an environment, or before it is used in one or the other context in the product or in the manufacturing processes, I think that's something we should consider. And again, it brings me back to the original point. We live in a biotope, and this biotope right now is our globe, our world. And I think that's what we take responsibility for. And I think the information that is available to us, we should use in a meaningful way. The regulator doesn't necessarily hammer on a gold standard that's derived from an animal system. And I'm calling it animal system because it's often not even a good model for human disease. As such, let's just be mindful and let's consider what our ultimate goal is going to be. It's not to get over the next 5 years without a lawsuit. It is to secure the future of humankind in the future, and I think that's what we all have in mind here.
Thank you very much. So let's take let's see, how much do we have for a break here? We have 15 minutes and come back at 10:25. And then we'll take up the topic of infusing regulatory considerations into research and development. So I hope some of our researchers in these systems will also come back and participate. Thank you.